Okay, so our first set of chapters that we're going to cover is going to deal with the cardiovascular system. We've got 3 chapters for this exam. It's going to start off with the blood, then we're going to move over to the heart, and we're going to finish up with all the blood vessels. Okay, so when we think about the cardiovascular system, it is just those 3 things. We got a pump, we got pipes, and we have the stuff that's flowing in the pipes. That's the blood. Okay, so from the standpoint of this cardiovascular system as a whole, we're going to be transporting a lot of stuff. Oxygen and carbon dioxide. We're also going to pick up nutrients from the digestive system and other organs and move them around the body. Hormones. This is where the immune system components are going to exist, and we're going to be able to transport them to places in the body that we need those. And we're going to be able to get rid of waste products. Okay. So, think about the blood. It is, if you remember, it is a specialized fluid, okay, and we consider it a connective tissue. It's a connective tissue because it meets those 3 characteristics of all connective tissues. We have different kinds of cells that live in an extracellular matrix, call it goo, and we've got protein fibers, okay? So those 3 things make it a connective tissue. Now, from function of blood, they're going to be very similar in a lot of cases to the functions of the cardiovascular system, because when we think about blood, it's going to be the thing that is transporting the gases, our oxygen from the lungs to the tissues, those nutrients, those hormones, those waste products, okay? We're going to filter that at the kidneys for the waste, okay? So we've got lots of the functions that we see for the cardiovascular system belonging to that of the blood, okay? But then we've got some other ones. We're going to regulate the pH and the ions that are inside the bloodstream. Okay, so calcium levels, potassium levels, sodium levels, things like that. We're also going to help neutralize acids. Okay, we've got lactic acid from the skeletal muscle that's going to be generated, so we're going to help to transport it to where we can convert it back to something useful. We're going to see the restriction of fluid losses at the injured sites. Basically, we're going to clot the blood. Okay, we're going to stop blood from leaking out of the body. So that's one of the functions here. We talked about the immune system function, the cardiovascular system. Well, that's here in the white blood cells of the bloodstream. Okay, we're going to move these around the body, make sure that they have plenty of the correct type of white blood cell to fight whatever infection it is that we might have. And we're going to stabilize the body temperature. We're going to use the blood, okay, and move it through the body in such a way that we can warm up certain parts of the body. We can cool down other parts of the body, okay? So we have a way to shunt the blood to places that we need it, okay? Okay, so when we have blood, this whole blood, this has just come right out of one of the vessels in our body, okay? It is composed of 2 parts, the plasma and the formed elements, okay? These formed elements are the cells and the cellular fragments that we see in the blood. We'll talk about red blood cells and white blood cells and platelets being these formed elements. The plasma is the fluid of the blood, okay? And it's this kind of yellowish to clear color that looks very similar to interstitial fluid. And in fact, it's what's going to be moved out of the bloodstream and form the interstitial fluid once it leaves the circulatory system. Likewise, interstitial fluid is also going to make its way back into the bloodstream. Okay, so that interstitial fluid leaks back in and becomes plasma once it's on the inside. So we're able to communicate the circulatory system to these cells that are outside of it, okay, where the interstitial fluid is. We're able to communicate this by sending this fluid across. We don't send the cellular components, though. We just send the fluid. But inside that fluid, we've got nutrients, we've got oxygen, we've got ions, we've got all kinds of things that we can move across, okay? Okay, so our types of formed elements. These are the cellular parts, okay? This is the red blood cells. Their job is to transport oxygen. That's their only job, transport oxygen. Second type of cell, white blood cells or leukocytes. Okay, erythrocytes, red blood cells, leukocytes, white blood cells. These are all part of the immune system. They are not, excuse me, they're not designed to transport oxygen. They're there to handle our immune system function. And then we've got platelets. Platelets are cell fragments that are involved in the actual clotting process. Okay, so we have stem cells that are in this bone marrow that are going to do a function called hemopoiesis. Okay, and hemopoiesis is how we produce these cellular components. So if you notice on this little diagram that we have at the bottom, there is stem cells that are called hemocytoblasts, and they can differentiate into two different stem cells, the lymphoidal stem cell and the myeloid stem cell. Well, the myeloid stem cell makes the bulk of these cells. It's able to make the basophils, the eosinophils, the neutrophils, the monocytes, the platelets, and the red blood cells, the erythrocytes. The lymphoidal stem cell, it only makes lymphocytes. And I think that shows us how different these lymphocytes are going to be. Okay, the myeloid stem cell can make all of these except the lymphocyte. It's so different. We can do a process to separate this blood. It's called fractionation. We use a machine called a centrifuge, and this centrifuge spins a sample of blood very, very quickly, and it allows those red blood cells to make their way to the bottom. The leukocytes then layer on top of the red blood cells. They're a little larger. And then on the very top, we see the platelets. And then that stuff that's on the inside, above those cellular components, we see that as plasma. So when we're looking at this, all of this portion here is the plasma, and then this is the formed elements. Okay, now when I was doing a lot of research on animals and things like that, a lot of times we would have to get DNA samples from their blood. So we would take their blood, okay, and one of the cool things is red blood cells, okay, they don't have any DNA in it because they don't have a nucleus. So we couldn't use any of these. We had to be able to get to this little band right here where the white blood cells are, because they're the only ones with a nucleus so that we could extract the DNA. Okay, some characteristics of blood. A little bit warmer than our normal body temperature, about 100.4 degrees Fahrenheit, okay, 38 C. Very viscous, it's thick, and it's a little bit alkaline, okay. It's on, it's close to neutral, but it's on the basic side of neutral, about 7.35 to 7.45. And we have to maintain this pH very strictly, okay. From a standpoint of the amount, it makes up about 7% of your body weight. So we're looking at, on average, adult males having somewhere between 5 to 6 liters of blood, where females, being smaller, having 4 to 5 liters of blood. And there's some other reasons for those differences as well that I'll talk about. Okay, so let's break this blood down into its different parts. Let's start with the plasma. Plasma makes up 50 to 60% of blood volume. And you can go back and look at that centrifuge slide where we had done fractionation. We wanted that plasma to represent 50 to 60%. Okay, when we look at the plasma, okay, that 50 to 60% that's in the blood sample, 90% of it, okay, actually more than 90% of that is water. Okay, and this kind of breaks it down for us. Okay, if you look at this portion here, it says 92% of the plasma is water. We've got 7% that are going to be the plasma proteins, and then we have this other group, these other solutes that are floating around. Okay, so when we look at the water, we know that water, from our chemistry lecture back in A&P 1, water is a wonderful substance. Okay, it holds temperature very well. It actually helps in chemical reactions and things like that. Okay, remember dehydration synthesis, remember hydrolysis, okay, remember hydrogen bonding, okay, all those things are going to be important as it is the main component of the blood. But then if we look at the plasma proteins, okay, that's this group that's up here at the top. Okay, we're going to have 4 different kinds. We're going to have the albumins, the globulins, and the fibrinogens. And then there is a group of regulatory proteins that make up less than 1% of this. Okay, and then we're going to talk about these other things that are inside the bloodstream. Okay, this portion that is the other solids, it's less than 1% or around 1%. We've got all kinds of electrolytes. Let's think about electrolytes. I think of Gatorade because it's full of a bunch of these different electrolytes that we can utilize. And these are things that have ionized once we put them into the water. Because remember, water was a good ionizer. Okay, it'll break those bonds and form ions. Okay, there's also nutrients. We picked up these nutrients from the digestive system and they are carried around our bodies. Okay, my wife has recently, because of West Nile, she became diabetic. Her body attacked her pancreas and shut down her insulin producing cells. So we have to monitor her blood glucose levels. Okay, so you can see here on this list that glucose is one of those things that we're carrying around inside the bloodstream. So if hers gets too high, she has a little pump that she can send a message to, and the pump will send more insulin into her body through the pump to reduce the amount of glucose. Okay, and if she gets too low, we have little glucose tablets that she takes to increase the amount of glucose in her bloodstream. And we'll talk about that with lots of things because there are amino acids there as well. Cholesterol's floating around. There's fatty acids that are in our bloodstream. Lots of things that we can utilize as a source, as that carbon, hydrogen, organic nutrient source to make ATP from. And likewise, because the cells are using these molecules, they're creating a waste. Okay, and that contributes to the amount of organic waste that is in the bloodstream as well. Okay, so those are those solids. So let's take a look at these. Okay, that extracellular fluid or interstitial fluid, okay, and plasma we've talked about. Okay, interstitial fluid, extracellular fluid, those two are the same thing. Okay, and you'll see it talked about both ways. Okay, the fluid that's outside the cell or the fluid that is bathing the cells. Okay, and the plasma, these are always exchanging things. Okay, the plasma and then the interstitial fluid exchange across the capillary walls. Now that's important right there. It only happens in capillaries. Okay, so when you think about the big aorta, we can't do it when it's in a blood vessel. It only happens when it is in a capillary. So in these big arteries and veins that we have in our body, we are not doing exchange there. We only do exchange in a capillary. Y'all don't get tired of me saying that, okay? So there is a difference then with what's going on between the plasma and the interstitial fluid, okay? So it says, number one, levels of O2 and CO2. So because the body is picking up, the cells of the body want the O2, we got to have more O2 in the plasma than there is in the interstitial fluid because we're going to utilize it, okay? Likewise, the cells have created more CO2. There's going to be more CO2 in the interstitial fluid than there is inside the bloodstream. And that's going to allow for us to go from high concentration, low concentration. Utilize that diffusion, that concentration gradient that we talked about in AMP1, okay, and load it up. Remember, both O2 and CO2 are lipid soluble, so that phospholipid bilayer does not create a barrier for this. It's able to go right through it, okay? And then we've got these dissolved proteins. The dissolved proteins do not pass through the capillary walls, okay? So that means inside the bloodstream, we're going to have a lot more of these dissolved proteins than we do out in the interstitial fluid. The way we feed the cells protein is through amino acids. We break them down so small that they can move those across that way, okay? So we're talking about these plasma proteins. Remember, it makes up about 7%. And of that 7%, 60% of them are albumins. That's the greatest number of the plasma proteins as far as a group goes. So we'll talk about albumins, globulins, and the fibrinogen, okay, and realize that fibrinogen is really low. Okay, so albumins, their job is to transport things that have a lipid component to them. So like fatty acids have a lipid component. Thyroid and steroid hormones are made from cholesterol. So we need something. These are lipid, and they'll stick together if we don't have a good substance to transport them with. Okay, so these albumins are going to help us transport lipids. Okay, fibrinogen, these are the ones that form the fibrin strands. Now, as fibrinogen, okay, it's soluble, meaning we can't see them. Okay, but if we need to clot our blood, okay, we go through a cascade of reactions that converts the fibrinogen into fibrin, and I'll teach y'all that toward the end of this chapter. Okay, now under certain conditions, we can remove, okay, the fibrinogen from the blood and give that to our patients as serum. A lot of times we want to give them serum if they're having problems with clotting. We don't want them to clot. Okay, they're clotting too much, so we're going to give them serum to dilute that. The last one is the globulins. Okay, and again, it makes about 35% of the plasma proteins, and two completely different types of globulins. One of them we called immunoglobulins. Another name for that is antibodies. Okay, immunoglobulins are antibodies. In fact, these immunoglobulins, a lot of times we'll learn the different forms, and they are the IgA, okay, the IgG. That means immunoglobulin A, immunoglobulin G, that kind of stuff, okay? So we're going to have these antibodies. Their job is to attack foreign bodies. And then we've got another set that are the transport globulins, and they're going to transport things, again, some of the lipids, and then also some of the minerals, okay, the metals that we have in our body, like iron. Okay, we've got some of these other ones up here, thyroid binding, because again, we have a lipid component to it. Steroid binding proteins, testosterone, things like that, lipid component to them. Okay, but most of the time when I think about globulins, my head first goes to antibodies, and then it goes to some of these other transport functions. Okay. And then we've got that group, that really small group that makes up 1%, okay? And what we're gonna see with this is it's gonna change a lot, because depending upon what we're doing, are we post-absorptive, okay? Have we absorbed all the food and we've got a ton of the nutrients in the bloodstream? Have we been fasting for a while? The hormones, what hormones are present? What time of the month is it? Okay, all kinds of things like that. Okay, even during the course of the day, we will see changes in this. Okay, now 90% of these plasma proteins are made in the liver. Okay, all of the albumins, okay, and all of the fibrinogen, most of the globulins, okay, but not the immunoglobulins. Those antibodies are made by cells of the immune system. We're going to call them plasma cells. Okay, it's a form of a lymphocyte. Also, there are a bunch of these hormones that are floating around. Those are made by our endocrine organs. Okay, and they also have that protein component to it that make up the plasma proteins. Okay, so let's take a look at these red blood cells. Red blood cells make up 99.9%, that's a bunch, y'all, 99.9% of the blood's formed elements. What does that tell you? A lot of red blood cells, right? A lot of these erythrocytes carrying a lot of oxygen, okay? One of the things that we need to keep in homeostasis in the body is how many red blood cells you got. So oftentimes it's measured. A lot of times when you go into the doctor's office, they may pull blood and just send away to see how much blood you have. And all the components that are in the blood can be analyzed. So different ways of looking at the numbers of red blood cells. We can send it through a machine that actually reports the number of red blood cells in a microliter of whole blood. How many of these red blood cells do you have? Quick and dirty way to do it is the way we did it earlier when we used the centrifuge and we spun it down and we looked at the packed cell volume, it was called. Okay, packed cell volume So when you centrifuge it and you look at it, and we want there to be around 40, 50% of the actual red blood cells making up the sample. So once it's spun down, we want to see about half of it being red blood cells. Okay. Normal red blood cell counts, males have more red blood cells than females do. Okay. Usually 4.5 to 6.3 million. Okay. In a microliter blood, females 4 to 5.5 million. Now, so looking at a hematocrit, that means males' hematocrit is going to be a little bit higher for the red blood cell than females are. And the difference is due to males produce the hormones, these are androgens, like testosterone, that actually stimulate more red blood cell production. Okay. The shape of this erythrocyte or red blood cell is a biconcaved disc. Okay, it's narrower in the middle than it is on the outside. So this is the thinnest part right here. It's not hollow, even though when you look at it under the microscope, it looks hollow. Okay, it's just like a somebody said a Smartie, the candy, a Smartie. Okay, which is concaved on both sides. Okay, why this interesting shape? Well, it gives you a bigger surface area. We've spent a lot of time talking about surface area. So the surface to volume ratio is bigger. That lets us absorb and release oxygen much more readily. They form kind of a poker chip, like a little disc, and we can stack these up and allow them to make their way through the narrow capillaries and then go through kind of single file order this way. And number three, it can bend. This shape allows for bending and flexing as they go through these small capillaries, so allowing them to pass readily and diffuse the maximum amount of oxygen possible. They live about 120 days. And by the time they are an adult, okay, when they are kicked out, we have removed the nucleus, the mitochondria, the ribosomes. They've made everything they're going to make. Their job now, it's been built. They float around for 120 days, and then we recycle them. Okay, the protein component that's inside the red blood cell, this is hemoglobin. Okay, and its job is to transport the respiratory gases. It is able to transport oxygen and to a lesser extent carbon dioxide. Okay, now this statement hopefully makes perfect sense. If males have more red blood cells than females do, we should also have more hemoglobin. Okay, since that's what makes up the red blood cell. So, and again, the numbers don't mean that much to me. Okay. Here is the structure of the hemoglobin molecule. So let's think about this. Inside of our red blood cell, in this little tiny piece right here, we're going to have a bunch of hemoglobin molecules. And the hemoglobin molecule is going to look like this. All of this, that's the kind of purplish blue color, that is the protein component. And then on the inside of that, we have a heme group, that's the yellow, and inside the heme is an iron. Okay. And what we do is we put 4 of these individual structures together. 1, 2, 3, 4, glob 4 proteins together, and that gives us the hemoglobin molecule. Okay, and it shows you here all of these hemoglobin molecules making up the red blood cell. Okay, if we attach oxygen to it, we call it oxyhemoglobin. If there's no oxygen to it, we call it deoxyhemoglobin. Okay. So, how many hemoglobin molecules in a red blood cell? Okay, that's a good question. Okay, there's 2 different kinds of hemoglobin we have. Okay, when we're little Cletus the fetuses and we are struggling to get oxygen because our respiratory system's not working, mom's breathing for us, and we're going to transfer the oxygen across the placenta, we need a form of hemoglobin that's stronger than mom's hemoglobin. Okay, that way we can strip the oxygen off of her red blood cells. And moms go through an increase in blood production just for this reason. Okay, they have more blood when they're pregnant than they do otherwise. So we need a form. This is called fetal hemoglobin, okay? And it's able to strip the oxygen off of mom's hemoglobin molecule, even though the blood isn't touching it, okay? The baby's blood is in a capillary, the mom's blood's in a capillary. Remember, the blood cells don't go across, but we're able to take the oxygen off, okay? It says it begins about the last 7 months in utero and continues over the first year of life, giving the baby a greater ability, okay, to absorb oxygen this way. And what we've been able to do, since this is a gene that would code for a particular protein, okay, and then once we get past a year, we're going to turn on a different gene to just make normal hemoglobin, okay? Since it's a gene, if we can stimulate that gene, we can help people that have problems with their red blood cells. So things like sickle cell anemia or thalassemia can be aided in us turning on that fetal hemoglobin molecule. Okay. Now, I asked the question a second ago, how many hemoglobin molecules in a red blood cell? Okay. Look at this number down here. Each red blood cell has how many? 280 million hemoglobin molecules. And each one of those hemoglobin molecules has 4 subunits, okay? And each one of those ions can bind an O2 molecule. So that means an individual hemoglobin molecule, because it has 4 of these, we can bind 4 O2 molecules to the 4 heme groups. That means 4 times 280 million is the potential that that red blood cell can transport of oxygen. That's over 1 billion molecules of oxygen. That's how important this oxygen is going to be for us. Okay, this hemoglobin can also bind carbon dioxide to it. Now, it doesn't bind at the same place that oxygen does, so there's no competition between carbon dioxide and oxygen for binding. Okay, the carbon dioxide actually binds to the alpha and beta chains, the actual protein part. Okay, but we produce something called carbaminohemoglobin. Okay, and we're able to move this back to the lungs, release the CO2, pick up more O2. Okay, anemia. This is a condition where we look at somebody's blood and they have too few red blood cells. Okay, so it's important to maintain homeostasis with respect to our blood. If we develop a leak where we are bleeding, okay, then we can't produce as many red blood cells that we need to, because we're losing them. We can run low on red blood cells. Okay, so anemia is a symptom that we have an issue somewhere. Okay, the lack of blood is due to some issue. So this is a symptom of it. And like it says, it can be caused by a number of different things. Okay, but when we think about anemia, what's the patient going to feel like if you don't get enough oxygen? You're going to feel very fatigued. Okay, very fatigued. So if maintaining homeostasis with respect to our red blood cells is important, we better have a good way to recycle them. And we do. Okay, we're going to, it says about 1% of our circulating red blood cells wear out every day. That's about 3 million red blood cells per second. So when we think about that, how many red blood cells do we need to make a second? Well, we better be making 3 million red blood cells or there's going to be a problem, right? So the way we do it is the macrophages that live in the spleen, the liver, and the bone marrow are going to help us with this. They're going to engulf the old red blood cells and destroy them, taking back the good stuff and getting rid of the bad stuff. Okay, so let's take a look at this. Here is a macrophage, and let's imagine it here in our liver, let's say, okay. And the bone marrow produced these red blood cells, so they've been out there for 120 days, okay, and it's time for them to go, okay. So here it is, 120 days old macrophage. Y'all remember phagocytosis? It's going to phagocytize this cell. It's going to bring it in. It's going to hit it with the lysosomes that are inside of the macrophage, and we're going to tear it down to its constituent parts. Now, if you remember the parts of the hemoglobin molecule, that's going to be the protein part, the heme group, and the iron. Okay, so all of the protein gets converted to amino acids. We put those in the bloodstream, and it gets carried all over the bloodstream, feeding the cells that need it, but also making its way back to the bone marrow so that we can use those amino acids to make more red blood cells. Okay, make more hemoglobin. Okay, the heme group we're going to do something else with, but let's go up with this iron. We're going to dissociate the iron from the heme, and here's the iron, and the macrophage releases it. And remember, there was the globulin fraction of our plasma proteins would carry minerals for us, okay, metals. So transferrin is one of those globulins that will do this, and it's going to transport that iron back to where it's going to be needed, and that's going to be to the bone marrow, okay? So we got the protein part going there, all the amino acids. We've got the iron going there. What we have left is the heme group. And this heme group, that little yellow part, little yellow disc that was on the inside, the macrophage converts it to biliverdin, which is kind of a green substance, and then it converts it to something called bilirubin, which is a yellow substance. And the liver uses that bilirubin to make bile. Okay, and bile is what we carry in our common bile duct, and we store this bile in our gallbladder. And when we eat a fatty meal, we release this bile down into the small intestine, and it makes its way into the large intestine, emulsifying the fats for us. And when we go to absorb the water that's in our large intestine, we get to take back some of this, okay, that we can utilize. The rest of it is eliminated in the feces. That's why we have this kind of yellowish color, yellow-brown color that we can see a lot of the time. Also, this bilirubin, okay, that is in the bloodstream, okay, can be excreted by the kidney, and that's what gives the yellow color that we see in our urine if we haven't drank enough water, okay? And if there's enough of this bilirubin, okay, that we can't handle the amount, we can actually store this in the skin cells, okay, the skin cells of the body. And we call that being jaundice, okay, when a patient is jaundice. I had a condition where I had hepatitis as a 17-year-old. Right before football season started, I contracted hepatitis. I got up and I went to the bathroom that morning, urinated, and it looked like black coffee. Okay, and I called for my mom, and we went to the doctor, did a urine sample there. He sent me to the emergency room for minutes into the hospital. And very soon after that, I became very ill. Felt fine, other than my urine was black. Okay, what had happened was my liver was fighting an infection. It wasn't able to handle this bilirubin, converting it to bile and that kind of stuff. So the excess bilirubin built up, okay, running all through my body, and my eyes, the whites of my eyes turned yellow. My skin took on a yellow hue, and that's what was being released here from the kidney. So much of this bilirubin that yellow color looked black. Cool, huh? Okay, so what we should never see, okay, in our urine is hemoglobin. Okay, if we do, we have a condition that we say is called hemoglobinuria. That means we have excessive breakdown of this. Okay, and because of that, we are breaking down blood cells in the urine. Okay, so finding hemoglobin in the urine often is a symptom that we've got an issue with blood cell breakage. Okay, blood cell lysing. If we find whole red blood cells, which again, this is much more common to find these, this is always a condition where if you find red blood cells in your urine, okay, that is always a trip to see your doctor, okay? I get that phone call, not as much as I used to, but used to, it was multiple times a year a student would call me and they would say, hey doc, I'm peeing blood, or I've got blood in my urine, or something like that, and what do I need to do? And I said, well, first of all, go to the doctor. And then their question was, well, what could it be? And there's a multitude of things, from really not any problem at all, to being very, very problematic. Okay. But it's one of those things that we do need to get checked out. Okay. So we've discussed this hemoglobin recycling, right? Biliverdin is the green substance that the heme is converted to once we take the iron off of it. Okay. All the amino acids get sent in the bloodstream, and we're just going to maintain a concentration of amino acids in the blood to handle the need to make more red blood cells. Okay, that iron is going to be recycled. We're going to send it through that transferrin molecule, which is a globulin fraction of one of our plasma proteins. Okay, the biliverdin is converted to bilirubin. Green goes to yellow, and then bilirubin can be produced, can make bile. And again, I said jaundice is if it builds up. Okay, and from that same bile, we get the same colorations that we see due to the intestinal bacteria converting this into the urobilinogens and the stercobilinogens, which give us this yellow coloration in our feces, and also because that bilirubin is going to be in the urinary system because it's going to make it into the bloodstream. Okay, we're going to have the same yellow coloration if we haven't taken in enough water that day. Okay, and we're trying to concentrate our urine. Okay, so we said the red blood cell lives 120 days. Okay, its life's about 120 days. And it takes us about 5 to 7 days to produce these red blood cells. Okay, it's going to occur in the red bone marrow. And remember that myeloid stem cell is the one that's responsible for the red blood cells, the erythrocytes. So this process is called erythropoiesis. Okay, erythropoiesis. So we've got that hemocytoblast that is in the bone marrow that can either go and turn into a myeloid stem cell. And with that one, we can make the red blood cells and most of the white blood cells. If it becomes a lymphoidal stem cell, we're just making lymphocytes. Okay. But we're going to start out with that myeloid stem cell producing a pro-erythrocyte. That's in day one. And notice there's a big nucleus here. And we go through the process with this nucleus of filling the cell full of hemoglobin. So the job is to use the genes that are inside the DNA to produce the hemoglobin molecule. Our poor little ribosomes are just busting out as much of the protein component as they can. When we get down here to day 4, notice that we're going to eject the nucleus. Okay, and what that does is leave us a structure called a reticulocyte. And this is what actually enters into the blood. It is not the nice, pretty shape that we see of a mature red blood cell, still kind of round, packed full of this hemoglobin. Okay, and then once it's out in the circulation, it converts itself to a mature red blood cell. So 5 to 7 days. Okay, remember we got to go from a myeloid stem cell all the way through that. I don't really care about these names. Reticulocyte is a good one because this tells us when we look at somebody's blood how many new red blood cells they're producing. If we see a high number of this, we know they're in the process of producing more blood cells. Okay, so as you know, all those things that we broke down to recycle, we need to build the red blood cell. So we need plenty of protein, amino acids to do this. We need iron. There's also some vitamins that are specific to this, vitamin B12, vitamin B6, and folic acid. And if we have limited amounts of any of these three, are we going to be able to build enough of the red blood cells? No. So what's that going to do to the number of blood cells that we have in the body? We're not going to have enough. Now, what's that called? That's called anemia, right? So there's lots of different forms of anemia. You can have iron deficient anemia where you're not getting enough iron in your diet, okay? You can have forms that deal with the vitamins, okay? Pernicious anemia is a specific type that deals with vitamin B12, okay? So a lot of patients that come in, they're low on blood, they're anemic, they're given a B12 shot. That's going to up the amount of B12. They're also given iron, okay? And hopefully we're making sure that they have ample amounts of this to produce the red blood cells that they need to. Okay. Now there's also a hormone that is very important in this process. The hormone is called erythropoietin. Okay, you may have heard it as EPO. Okay, it makes the news quite a bit when they're talking about blood doping with things like the Tour de France or the Olympics. It comes up with the Olympics a lot and that kind of stuff. Now, this hormone is produced in the kidneys, and the kidneys have cells that they are going to respond to when we have low amounts of oxygen, okay, in the peripheral tissues, which means we don't have enough red blood cells delivering enough oxygen to those tissues. We call that hypoxia, if you remember, okay? Now, if we don't have enough red blood cells delivering it, that means we need more red blood cells, right? But there are also things like diseases that would cause you to become anemic, so we would have more erythropoietin produced. Also, if we go to altitude, okay, if we go to altitude, it's harder for us to breathe. If you've ever been up in the mountains, if you've ever been at Denver or someplace like that, it is very hard to get enough oxygen in. It's not that there's not enough oxygen there. It's that the pressure is too little for us to ventilate the amount of oxygen that's there. Okay, so what a lot of athletes do is they go travel to places that are high altitude and they do their training there. So if you're training while you're at altitude, you're going to produce more red blood cells than the person that is here in Denton, Texas, training the same amount. So if you've got more red blood cells, can you stay oxygenated longer during the race? Yes, you're going to have more available oxygen. You're going to be able to stay aerobic longer, and that's going to give you an advantage because you're going to be able to produce a lot more ATP under aerobic conditions than you can under anaerobic conditions. Okay? So, when my son went to Mount Everest, to climb Mount Everest, okay? And he was going to climb the whole thing, but he did want to go to the base camp, the first base camp on Mount Everest. And when he arrived, okay, they wouldn't let him immediately go up on the mountain. You had to acclimate, okay, for about 7 days. And if you think back, how long does it take to make enough red blood cells? To make red blood cells, about 7 days, okay? So after 7 days, he had more red blood cells. He was more fit to climb to that first base camp, okay? So lots of different tests, okay? And if you're looking, when we're low, here's the depressed levels here. All of them basically point toward anemia, whether we're dealing with hematocrit, how many reticulocytes you got, what's your hemoglobin concentration, what's your red blood cell count. All of those are saying, okay, we don't have enough. All of these over here say, okay, we've got too many, okay, if they're elevated, okay? And a lot of times this gives us information. If the patient was in a wreck and lysed a lot of red blood cells, took specific drugs that are known to lyse red blood cells, and we find a bunch of reticulocytes where the patient has reticulocytosis, okay, we can trace that back to an event, okay? Okay, these red blood cells have one other thing going on that we need to talk about, and that is that they possess proteins on the outside of their cell. Now, we talked about this back in A&P1. We talked about that phospholipid bilayer and having carbohydrates and proteins on the outside that would allow for identification of cells by our immune system, okay? That would tell us, okay, these are our cells. We don't want to mess with those. Those are good cells, okay? So the immune system normally is set up so that it targets non-self cells. So normal cells that belong to us are ignored. It's the foreign one, those that are not part of our cells, non-self cells, they call it, okay? So we do the same thing with these red blood cells. These red blood cells have proteins on the outside of them that allow us to recognize these are our blood cells, okay? We call these antigens, these proteins on the outside. And basically an antigen is anything that can cause an immune response, okay? And these are going to be genetically determined. In fact, this was some of the first bits of genetic paternity testing that we could do, because we could look at two different parents and we could figure out what the child's blood type was, okay? And we could compare that to the parents, and sometimes we could exclude the male. Usually it was the male because, and we called it paternity testing, because the female was the one that was pregnant. So it was easy to figure out that she was involved, okay, in the maternal side of the genetics. It's harder to identify who the dad was. If you've ever seen the Jerry Springer show, you know that firsthand, okay? So this is one of the ways that we could look at to determine whether paternity, we could exclude somebody from being the father. We'll say it that way, okay? So what we're looking for is the presence or the absence of these antigens on the surface of red blood cells. And you can have an A antigen, you can have a B antigen, okay? And there is an RH antigen that we'll talk about. So we got A antigens, B antigens, and RH antigens, okay? So if, let's say, you have these little round red antigens on the outside of your red blood cells, these guys right here. This is the A antigen, okay? That you're a holder of the A antigen. If you have the little blue triangles, then you are a holder of the B antigen, which means if you have the round ones and the blue ones, then you're a holder of both A and B antigens. And if you got nothing around you, you don't have any of the antigens, okay? None of the antigens means you're type O, okay? Now there's another step to this that we need to talk about, okay? If you are type A, you don't make antibodies to type A blood, because then you would attack yourself. You make antibodies to type B. So these antibodies that you make are called anti-B antibodies. And notice they got the little triangular receptor here that would bind to these guys very easily, okay? They would bind to those, okay? If you have blood type B, then you make antibodies to blood type A. Notice you got the little cup here on the end of our antibody, and that's going to fit perfectly onto this, and they're going to bind, okay? Now, if you got both of these, you can't make any of the antibodies, because that's all we got, okay? If you have type O, you don't have either one. You don't have any of the round ones. You don't have any of the triangles, okay? So you make antibodies to both A and B, okay? Make antibodies to both A and B. So this is how we do it. Patient comes in and we're going to check their blood type. And we could do this here in lab. We have these little lancets that we poke your finger with, and we'd make you give us some of your blood. We'd mix it with antibodies, see which one you had, okay? So let's think about it. If here's our blood sample, and we mix it with anti-A antibodies, that's these guys right here. Those are the ones with the cup. They're going to target the A protein, the A antigen. And that means they would clump. So notice how it clumps here. Now these would not target these because they don't fit. So it would not clump for B. Okay, let's try this next one down here. Here we got our blood sample. We're going to check the anti-A, and they don't clump, so they don't have the A, but they do have the B. We got clumping with the B, so we know that this is blood type B. Here's us a new blood type. We're checking, and we get clumping with A, we get clumping with B. We know they got both A and B. And then we got our last blood type here that we're checking, and we got no clumping here and no clumping here. Okay, so they don't have the A, they don't have the B, so we know they are O. Now, the other one that we're looking for is the RH factor, and this is called the anti-D antigen, the RH factor. You hear it called both ways, okay? And the RH comes from rhesus monkeys. It was found in rhesus monkeys first, okay? So if we hit them with the Rh antibodies and they clump, that means you're positive for the Rh antibodies. That's where the little positive sign comes from. So this person would be A positive. This person would be B positive. This one would be AB positive. We got clump with everything. No clumping on this one, no clumping on any of them. That would be O negative. Okay? And this is real easy to do. Like I said, it takes a matter of minutes. Okay? And we can identify a person's blood type. Okay? Right there in the emergency room. Okay? Now, there's a nice thing. This next is the exact same slide that we just looked at. It's just a different picture. Okay? It's just a different picture. It does say that these antibodies are going to stick to the proteins. We call that agglutination. They're going to agglutinate with it. They're going to stick to it. Okay? So that's what we're looking at. You hear a lot about agglutination, and you'll hear about more of that in micro. Okay? So this is a table that shows you how blood can be donated. Okay? This is your blood type up here. Okay? So if you're blood type A, you can get blood type A. Okay? You can't get blood type B because it's got antibodies. Okay? B has antibodies that will react to your blood. Okay? AB, okay? Antibodies A and B. Okay? But we can give O because there's no antigens, so there's no reaction that's going to be made. Okay? So here's blood type B. Okay? If you're blood type B, you can't be given A. You can be given B. You can't be given AB, but O's okay because no antigens on the O. If you're blood type AB, okay, we're okay with that one because A and A. B we're okay with because both of them have that. AB, that's a perfect match. And O has no antigens on it, so no antibodies would attach to it. Okay? If you're blood type O, okay, your blood type is O, you can't get A, can't get B, can't get AB, but you can get O. And what that tells us is, if we look at this, is because O has no antigens on it, we can give it to anybody because it's not going to react. The antibodies that you might have will not react with it, so O is called the universal donor. Okay? If you have blood type AB, okay, you're not going to produce any antibodies, so you can be given any blood type. So these are the two. So blood type AB is the universal recipient, okay? Blood type O is the universal donor. And then we have to figure in whether it's positive or negative, okay? It's positive or negative. We don't want to screw that up either. Okay, we can screw that up. And back in the day, okay, I didn't have to have a blood test, so your parents probably didn't have to have a blood test, but your grandparents may have had to have a blood test when they got married. Okay, they were looking at this Rh factor, okay, because there's a problem with the Rh factor, but it's a little different than it is with the ABO system that we were just talking about, okay? You have to become sensitized, okay, to Rh positive blood. So a person's either Rh positive or Rh negative, okay? And if you are Rh negative, okay, and you are given Rh positive blood, we will make antibodies to that protein because that's a foreign protein. We'll make antibodies to it. But here's the deal. It happens the way that like the flu does. You have to see it first before we can make these antibodies. Unlike the ABO system, okay, where we're born with these antibodies. If you have blood type A, you are born with B antibodies. If you're blood type B, you're born with A antibodies, that kind of thing. This one, you have to see it first. So here's the deal. If you're an Rh positive mom, that means you have the protein, so you're not going to make the antibodies to it, right? So if you have an Rh negative baby, that's not a problem. If dad's Rh negative, mom's Rh positive, dad gives the Rh factor to the baby, we get no problems. Okay? But if the mom's Rh negative and the baby daddy is Rh positive, and the baby's Rh positive, here's the deal. Here's the Rh factor right here. Okay? The first pregnancy, this is not a big deal. Okay? But there's a problem. When the baby's born, baby's Rh positive blood is going to mix with mom's, okay? And mom is going to make antibodies to Rh because she's Rh negative. She's seen Rh positive blood. She knows it's foreign. It's been in her body. She's going to make antibodies to it, okay? So now the second baby that's born, and if that baby is Rh positive, okay, these antibodies that mom has made can move across the capillaries. Remember, the blood doesn't, the blood cells don't mix, but the proteins, the antibodies can. So the Rh antibodies go across into baby's blood and it starts attacking baby's blood. Okay, and it can cause death. Okay, it's known as hemolytic disease of the newborn, or sometimes you hear it erythroblastalis fetalis. Okay, if the baby's born alive, a lot of times it is anemic and jaundice. The jaundice comes in from all of the hemoglobin that it's recycling because it keeps destroying all those red blood cells because of those antibodies. Okay, so the baby may have to have a complete transfusion. Now, we came up with a drug. The drug is called RhoGAM, and moms are given RhoGAM, okay, if you're Rh negative. Okay, so if you go in, you're Rh negative at about the last 3 months of pregnancy before delivery, RhoGAM is going to be delivered. That way, if you have an Rh positive baby because you're Rh negative, you won't make antibodies to the Rh positive blood. And that's why we don't have to have blood tests anymore. Okay, because we figured out a way around it. What happens is this cross reaction. In this case, you can see that we have antibodies that will target the B antigen. And here is a bunch of cells with the B antigen on it. And these guys bond directly to those. Okay, they cause them to clump. That's agglutination. And they cause them to burst. That's called hemolysis. And that's going to dump all that hemoglobin and stuff. Remember, 280 million hemoglobin molecules per red blood cell. It's going to be a lot of mess to clean up. Okay. Okay, we've got the red blood cells, the erythrocytes taken care of. Let's move on to the white blood cells, or the leukocytes. Okay, these guys are strictly immunological cells. These are going to be part of the immune system. They do not have any hemoglobin in them. Their job has nothing to do with carrying oxygen. These are nucleated. They've got all the other organelles that we think about. And instead of carrying the oxygen with that hemoglobin molecule, their job is to defend the body against pathogens. They're also going to be there to help remove the toxins and the waste. And they help us attacking abnormal cells, those that are infected by viruses and those that are cancerous. Hopefully we will find all of those. The white blood cells will find those and they'll destroy our own cells. Okay, so when we think about these white blood cells, they are going to be able to leave the bloodstream, unlike the red blood cells where we think about them existing just inside of the cardiovascular system. White blood cells are going to be able to move out in the connective tissue. They're going to be able to be put into the lymphatic organs that are outside of our circulatory system. So really we see small numbers actually in the bloodstream. There's a lot more red blood cells out there than there are white blood cells. And we're going to see 6,000 to 9,000 white blood cells per microliter. And there is some homeostasis that needs to occur there. You don't want to have too few of these and you don't want to have too many of these, okay? So we said they're circulating. They're going to be able to migrate out of the bloodstream. We can call that emigration, or you can call it diapedesis. But they're able to do something called margination, where the activated white blood cell can adhere to the vessel wall and squeeze through at the level of a capillary. So they're able to have this kind of movement that's called amoeboid movement, where they're able to kind of squish themselves through things. If you remember, if you ever watched an amoeba move, it does require ATP, does require calcium in order for us to change the shape of this cell and allow for this emigration or diapedesis. The third thing we should think about is that these are attracted to specific chemicals. Okay, they have something called positive chemotaxis, which means if chemicals are released in the body, they will draw white blood cells to them. Okay, so if we get damaged tissues, that is a chance for pathogens to make their way into the body. We need to have some white blood cells over there, making sure that we knock them out if they make their way into the body. And they have different ways of killing things. Some of these are going to be phagocytic. And if you remember phagocytosis, they're able to engulf large things. Some are more small things like the neutrophils, the eosinophils we'll talk about. These are called macrophages. They're smaller. But the monocyte, it is a macrophage when it grows up, and it can consume large quantities of viruses, bacteria, things like that, as well as waste and pathogens. Okay, we've talked about those red blood cells. Now let's get to these white blood cells. Okay, here's what they look like. Top left, neutrophil. Middle one, top, eosinophil. One on the right, that's the basophil. Then we've got a monocyte, and we've got a lymphocyte. Now, we're going to have to be able to identify these, okay? Use your red blood cells that you can see there as a standard to compare the size to, because red blood cells are all about the same size. The top row, as you go across, they all are what I would consider the medium-sized white blood cells. You can fit anywhere from, I would say, about 3 of those inside of those white blood cells. Okay, when you get down to the monocyte, monocyte's the biggest. Okay, you can fit 4 or 5 sometimes in a monocyte. And then when you look at the lymphocyte, that lymphocyte is the smallest. We may only be able to get 1.5 to 2 sometimes in a lymphocyte. So size is going to help us out a lot. And then notice that there's some granules here. We can see some different colors to them. The neutrophil has some kind of purple staining granules that you can see readily. The eosinophil has red granules. The basophil has purple granules, and it's completely full. It's to the point where we can't even see the nucleus on this basophil. The monocyte really doesn't have any granules at all, and the lymphocyte, it doesn't have granules. Okay, it's small. When we look at the cytoplasm, notice that in the neutrophil, the monocyte, and the lymphocyte, we do get to see a little bit of cytoplasm. And the cytoplasm takes on different looks because the granules that are there in the neutrophil, it looks very speckled. The monocyte has a good cytoplasm. A lot of times it kind of has a bluish hue to it. You can kind of see that here. Some of the pictures that I'll show you, I'll try to point that out as well. And in the lymphocyte, it has just a tiny bit of cytoplasm. Okay, and we say that encircles the nucleus. We call that the cytoplasmic halo of a lymphocyte. Okay, also the shape of the nucleus. Notice that the neutrophil has a bunch of different kind of bulbous lobes to it. If it has 3, 4, 5 of those, you automatically know it's a lymphocyte. Or excuse me, it's a neutrophil. The eosinophil has 2, and they're shaped a lot like earmuffs, headphones, that kind of thing. There is a little band that goes across the top between those 2. I'll try to point it out to you here with my little marker. It actually is this part that's going across right here. Okay, so a lot of times that's what you'll be looking for. Okay, the basophil, like I said, you can't see the nucleus in the basophil on our slides. And then the monocyte typically has a C-shaped, a kidney bean-shaped nucleus. Okay, so those are the way we're going to tell them apart. Oh, and lastly, I should go ahead and point out we do have some platelets here as well. Talk about the platelets coming up, and in a lot of the slides that you will be looking at, you'll be able to see these little bitty platelets. Okay, first one we're going to talk about, the neutrophil. As you can see, blobby, lobed nucleus, all touching each other. excuse me, we call it a polymorphonuclear leukocyte because that nucleus is polymorphic, meaning it has a lot of different shapes to it. Okay, these neutrophils are the most abundant that we're going to find in the bloodstream. Okay, 50 to 70% of the circulating white blood cells that we're going to encounter are neutrophils. So when you look under a microscope and you're looking for these, usually that's the first one we're going to identify. Okay, it's easy to identify. We got those little granules, we got the multi-lobed nucleus, and it's medium sized. Okay, these guys live about 10 hours. They eat anywhere from 12 to 24 bacteria usually. Like it says, very dense 2 to 5 segmented nucleus with pale cytoplasmic granules inside of them that contain lysosomal enzymes and bactericides like hydrogen peroxide and superoxide that are good at destroying bacteria. Okay. We consider these kind of the most ubiquitous and generalist. Okay, they can attack bacteria, fungi, viruses. They're very active. We got a ton of them out there. They're kind of our the go-to ones. Let's see if we can get these knocked out. Okay, they can engulf pathogens. They can digest these pathogens. They can release the granules that are there. They also release hormones, call for other cells, and these are going to help contribute to local inflammation, which means they can release chemicals that can cause redness and swelling in the area. Okay, a lot of times when we find pus, when we're associating it with an infection in this particular area, this is what we're finding is all the dead neutrophils and the stuff that they have been cleaning up. So we get cellular debris, waste, and the dead neutrophils because they only live, about 10 hours. So if we're really fighting a fight there, we can have a buildup of these. Eosinophil, okay? Eosinophil, also about the same size as the neutrophil. Red granules, headphone-shaped nucleus. This one is very important in allergies, okay? Allergies as well as parasites, okay? They're called acidophils because those granules, when we stain them with Wright's stain, which is a pH stain, it causes the acids to stain red, okay? Now this represents only about 2 to 4% of the circulating white blood cells. 2 to 4%, that's not a lot. That means we're going to look at about 100 of these, maybe 50 or so before we'll ever find one of these. And then we've got to find a good one. So a lot of times we spend a great deal of time in lab looking for eosinophils, okay? Bilobe nucleus, they're able to degranulate. They're able to release these granules, and that helps them attack these parasites. It's kind of like pouring salt on a slug, okay? I did say sensitive to allergens can help control the inflammation with enzymes that counteract our inflammatory response. Basophil. This is the one that occurs in the least number. Less than 1% of our circulating white blood cells are basophils. So these take a long time to find. When we find them, they look very grainy. They're medium sized, okay? And they're able to also degranulate, okay? They have histamines and heparin in here, and what that's going to do is help to dilate the blood vessels. That's going to bring more blood into the area, and it's going to prevent it from clotting. So we're going to get an enhancement of this redness and swelling that we're going to see, okay? When you dilate blood cells or blood vessels, that means more blood into the area. That means more fluid into the area, so we get redness and swelling there. Then we've got the monocyte. This is the largest of our white blood cells. Notice it's kind of amoeba shaped. It's not round like the other ones have been. This one takes on kind of an amoeba shape. And then inside of it, the nucleus, okay, is kind of purplish with a bluish tint to the cytoplasm, okay? Big C-shaped nucleus or kidney bean shaped nucleus. Definitely the largest. And this one reaches adulthood once it leaves the bloodstream, okay? After it exits the bloodstream, it becomes a macrophage. And then it is a big eater. It will consume tremendous numbers of bacteria, okay? It's got those lysozymes or lysosomes inside that can tear them down once we move them into the macrophage. Okay. They also can secrete substances that attract immune system cells. We'll talk about that. We'll get to the immune chapter. And also fibroblast cells, which will come in and start cleaning up all the connective tissue damage and things like that we've got. Okay, the lymphocytes, these are the smallest of our white blood cells. And when you look at that, it's just a little bit larger than these red blood cells beside it. Okay. Also, we can see that little cytoplasmic halo forming around this ring right here. These are quite plentiful. Anywhere from 20 to 30% of your circulating white blood cells should be lymphocytes. Okay. They have a big round nucleus with a thin cytoplasmic halo. Okay. They're able to go in and out of the bloodstream. We'll talk about this in the immune system and the lymphatic system as well. So we find a bunch of them in connective tissue. We find a bunch of them in lymphatic organs like lymph nodes and things like that. They are part of the body's specific defense system, meaning we're going to identify who it is and we're going to put cells out there that know how to kill that particular strain of whatever it is. Okay. We got T cells, B cells, and natural killer cells are the three different classes. And we'll spend a lot more time learning about these in the immune system, but let's just get a generic idea of what's going on here. T cells are part of our cell-mediated immunity, meaning they attack foreign cells directly. Okay, so T cells go out and destroy other cells. Okay, now, these are also going to be important in that they are able to coordinate the immune system. So they're able to communicate actually to these other cells that are called the B cells. And what B cells do is produce antibodies. Okay, part of the humoral response, and they're going to distribute these antibodies. Specifically, the plasma cell is going to make these. Okay, make these antibodies for us. But there are different forms of this B cell. So this side is killing cells directly. This side is making antibodies that will go out and tag things for destruction. And then we have the natural killer cells that will attack our own tissue, abnormal tissue cells that are ours, like it is cancerous, like it is infected by a virus or something like that. They're able to kill those. So we've got some disorders with respect to these white blood cells. If you have too few white blood cells, now you're not able to turn on your immune system as well as it should. We call that leukopenia. If it's too high, we call it leukocytosis. Okay, so somebody that is coming off of a infection or something like that, they may have a abnormally high count, so we would be able to identify that they had recently been sick, and we're maybe in the progress of shutting down this immune response. If it is extremely high, we call that leukemia, and you hear a lot about that in the medical field and stuff like that, and we'll talk about it. Picture down here shows us these all come from the hemocytoblast. Okay, here's our stem cell hemocytoblast. Remember that it can either go the myeloid stem cell route or the lymphoidal stem cell route. If it goes the lymphoidal stem cell route, we're going to make the lymphocyte. Everybody else comes from this myeloid stem cell. We get the red blood cells. We get the white blood cells. Okay, all of these come from the myeloid stem cell. So the macrophage, the neutrophil, the eosinophil, the basophil, okay, all of that comes from a myeloid stem cell. The one we haven't talked about yet is the platelets, okay, and that's this group right here. So that's the only one that we have left to talk about with respect to the formed elements, the cellular components of the blood. Okay, so again, myeloid stem cell produces all blood cells with the exception of lymphocytes. The lymphoidal stem cell only produces lymphocytes. Okay? And remember, these are hanging out in the bone marrow. Okay? That's where these stem cells are going to occur. Okay? And all of these are going to develop fully so that they look like what they're supposed to, with the exception of the monocyte. Monocyte leaves, okay, floats around and becomes mature once it enters our peripheral tissues. So while it's not in the bloodstream. Okay, and this just says lymphoidal tissue. We've got lots of it that we'll talk about where these lymphocytes are going to hang out. The thymus, the spleen, and the lymph nodes are havens for lymphocytes to chill out and be ready to fight the fight. So here's a good table that talks about the red blood cells. It tells you how many we got, what they look like, what their functions are, and any interesting remarks about that. Okay, so it's a good one to look at. And questions always, do I need to know abundance? Yes, we need to know abundance of these, how many we should find, what's normal for our white blood cells. Okay, you're going to be able to identify them because I want to have you figure that out with pictures and that kind of stuff. Okay, do you need another function? Of course, that's good for our lecture exam. And then this is also important stuff, okay, kind of the history of what this cell does, the physiology of that cell, that kind of stuff. Okay, that brings us to the platelets. Okay, platelets, little bitty sandbags out here is what I like to think about a platelet. It has a cell membrane around it. Okay, and it is the cytoplasm from a huge cell. All of these guys are platelets here, and their job is to stop the bleeding, help clot the blood, and stop the blood from leaking out of our bodies. Also known as thrombocytes. So a lot of the terms become things like thrombocytopenia or thrombocytosis. Too little, too many. Always penia, too little. Cytosis, abnormally high. Okay. These platelets, once they're released, they're going to circulate in the body 9 to 12 days. Spleen helps take those back when we want to recycle them. Okay, the dead ones get phagocytized in the spleen. If we don't have a spleen, the liver will take over for that. Okay, and then we're going to hold about 2/3 of these in reserve. Okay, that way that if we need it, let's say we were in an automobile accident and there were lots of internal bleeding, we would need lots of platelets in order to help stop the clot or help stop the blood loss. So our functions for platelets are going to be, they're important in releasing clotting chemicals. They're part of the hemostasis, us stopping the bleeding. They form a temporary patch. Like I said, they're like sandbags. If it's flooding, we're going to throw sandbags down. Okay, and they're able to actively contract, so they can help pull the wound together once we have stopped the clot or the blood from leaking out, and we form that clot. Okay, so making platelets, we've talked about poiesis before, the formation of, the creation of, this would be the creation of platelets. Okay, again, in the bone marrow, that's where our stem cells are. And that myeloid stem cell produces a cell called a megakaryocyte. Megakaryocyte is what produces the platelets. Okay, each megakaryocyte can produce about 4,000 platelets. And you can see these kind of budding off. Imagine us just doing exocytosis with this, okay, and pinching these little bitty platelets off of the cell membrane, okay, and you can see all of these are being created by this megakaryocyte. Okay, this next bit is what I normally would ask you on an essay question. If we were in class, the question would be, explain hemostasis, which is how we stop blood loss from the body, okay, how we end the bleeding. And it occurs in 3 phases: the vascular phase, the platelet phase, and the coagulation phase. And these phases are not the normal way we think about it, where the vascular phase runs and then it ends, and then the platelet phase picks up and then it runs and then it ends, and then the coagulation phase starts and then it runs and then it ends. Basically, all of these are going to happen one after the other, but they're going to continue to run, okay? So the vascular phase is going to start and it's going to continue, and then the platelet phase is going to start, and and then the coagulation phase starts while all of this is going on, okay? So let's take a look at this, okay? First phase, vascular phase. When we cut through a blood vessel, as you can see here, when we cut through this blood vessel, first thing that happens, we get a 30-minute contraction. It's called a vascular spasm, okay? This vascular spasm basically contracts that blood vessel, okay? We've talked about dilating the blood vessels. This is going to be the opposite of that, okay? And it's going to pinch it down, and that's going to reduce the amount of blood loss because we're dialing down the diameter of that blood vessel, okay? So the way it works is the endothelial cells, those are the epithelial cells, but the ones that are on the inside next to the blood in the vessel, okay? They're going to contract, and because of this cut, they are going to release chemicals, okay? They're going to release chemical factors and hormones that are going to affect what's going on in that specific area, okay? Now, these endothelial hormones are going to stimulate smooth muscle contraction, and they're going to say, okay, let's start increasing cell division in this area. And that way, we'll get repair going on, okay? The endothelial cells, when they have released some of these other, like tissue factor, that's going to stimulate cell division, prostacyclins, that's going to work on dialing down our blood vessels and things like that. And these endothelial cells are going to become sticky. Okay, they're going to release a compound that is going to make them sticky, and that makes them it makes it easier for us to seal off this blood flow. Okay, so now that we've got that going on, the platelet phase is going to pick up. And the reason the platelet phase occurs is because of the chemicals that are released by those endothelial cells, okay? And the platelet phase is going to begin about 15 seconds after the injury. So we initiate that vascular phase, and then 15 seconds later, we're going to crank up the platelet phase, okay? And the platelets are going to become activated. The chemicals that are released by the endothelial cells activate the platelets in that area, and these platelets, okay, grow little fingers where they were these little sandbags. Now they're going to have lots of little pointy processes that stick out, okay, increasing their surface area. And it's going to cause them to get sticky too, okay? So the sticky platelets are going to stick to the sticky cells that were created by the cut, okay? So we got platelets sticking onto the cells, okay? As these platelets continue to stick to the cells, it forms something called a platelet plug, okay? And this platelet plug, if it's a small enough type of a cut, this is enough a lot of times to close the small breaks, okay? And you can see it happening here. Here, we've got those sticky cells that are going to be along the edge here, okay? And they're going to start, the platelets are going to start to stick to them. And since the platelets are sticky, they're going to stick to each other. And basically, we're going to form this plug right here due to platelet aggregation. Okay, so because the endothelial cells turned on, activated those platelets, now these activated platelets start releasing chemicals of their own. They're going to start to release clotting factors as well as growth factor, okay, so that we can, again, fix repair what's going on, and initiate this clotting. Okay, and then we got some other ones that are important in the actual processing of the clot and that kind of thing. Okay, so now that we've got the platelets doing their job and we've covered up the endothelial cells, we start to see prostacyclin released. Okay, and that's going to inhibit that platelet aggregation. Okay, so prostacyclin released by the endothelial cells says, okay, that's enough. We don't want this thing to continue to grow out of hand. Okay, and like the prostacyclin that's released, there are some other inhibitory chemicals that can be released by the white blood cells that are there that says once we get it where we're not losing any more blood, we need to stop this. Okay, and we see that like circulating enzymes that are inside the bloodstream. Serotonin is released here, and that forms a negative feedback on the size of this platelet plug. Says, okay, we need to stop this. Okay, stop it. Don't continue to grow. We've isolated the area. We've got a clot in that area. We've stopped the flow of blood outside the body. It's not leaving the body anymore. And then we begin the coagulation phase. Okay, about 30 seconds or more, depending upon the size of the damage that's done to the vessel, size of the vessel, and that kind of stuff. We're going to start the actual blood clotting. We call this coagulation. Okay, and there's a bunch of steps that's involved in this, and it is a cascade of reactions, chemical reactions that have to go. But y'all remember that we've got the fibrinogen as a plasma protein circulating in our plasma. Okay, it's in the bloodstream. All we've got to convert it into a form of a protein that's called fibrin. And what fibrin is, okay, it's insoluble. That fibrinogen, remember we said it was soluble, we couldn't see it. But the fibrin is insoluble, and it basically is like hair, okay? And if you've ever had a clog in your shower or something like that, my wife calls me in every once in a while and says, the drain in the shower is not running very well, okay? And I know that there's a bunch of hair in there. And if you've seen my head, you know that's not coming from my head. My wife has long hair, long curly hair, and it traps all kinds of particles in there and eventually makes it hard for the water to escape. That's exactly what we're doing here with the coagulation phase. We're reinforcing those platelets with these proteins, and we're forming a big old hair clog, okay? The goal is help cover and infiltrate that platelet plug, trap the blood cells that are there, and seal off the area. Okay, so let's say, and I do this quite a bit since I shave my head, I nick the top of my head or the side of my head or the back of my head, and it bleeds. Okay, and what I do is I go over and give you a piece of toilet paper, and I start blotting that spot. Okay, it's not going to bleed for long, so I blot it. It's nice and red right now. Blood's red on my toilet paper, and as I continue to blot it, it gets less and less red. It goes from red to kind of a pinkish color. And then from a pinkish color, it turns into this kind of yellowish color. Okay. And then it dries up. Put a Band-Aid on it or something like that. Okay. So what's happened is, cut my head. We had to go through the process of making it sticky, getting that platelet plug put in place, and as it's forming, I'm starting to slow down the amount of actual blood cells that are leaving my wound. Okay, that's why it's going from red, where there's plenty of them coming out, to now it's slowed down, it's pink, and then we've stopped them all. Now it's yellow. The only thing that's leaking out is the plasma. Okay, it's the plasma that's coming out. That's what the yellow substance is. Okay, and then eventually we stop the plasma from leaking out, just like that hair clog does in the shower. Okay, so let's look at how it works. Okay, so like I said, it is a cascade of reactions. It's going to involve 3 different pathways. And back in AMP1, we talked about extrinsic regulation and intrinsic regulation. And we said intrinsic regulation happens right at the location. Okay, so if we're clotting the blood, that's going to be inside the bloodstream. That's the intrinsic pathway. It's going to come from the actual bloodstream. The extrinsic pathway, that's coming from further away. It's going to be outside the bloodstream. Okay, so we've got three different pathways, one involving the extrinsic pathway, the other involving the intrinsic pathway. So the two come together and form the third pathway, which is called the common pathway. It says that's where the intrinsic and extrinsic pathways converge. Okay, so what we're going to do for this extrinsic pathway, remember it's outside the bloodstream, it's going to come from the damaged cells that are releasing the tissue factor. Okay, this tissue factor and other compounds activates factor 10. Okay, that's Roman numeral 10, factor 10. Okay, the intrinsic pathway coming from inside the bloodstream, remember the platelets were releasing chemicals too. So those platelets, here's platelet releasing factor 3 is a good example of one that's released, and that's going to do the same thing, and it's going to activate factor 10. Okay, so we've got extrinsic pathway coming from the damaged cells. We got the intrinsic pathway coming from the blood itself, those platelets, and they're going to converge here, and that's where factor 10 is. Okay, factor 10 right here. And then from factor 10, we're going to take it through the common pathway. Common pathway says once you've activated factor 10 forms an enzyme called prothrombinase. Remember, ASE on the end of anything is an enzyme, and pro means before, so we're going to convert prothrombin into thrombin. Okay, and once you get thrombin made in the bloodstream, thrombin is what is able to convert that fibrinogen into the hairs that we want to have of fibrin. Okay, so that means from the extrinsic pathway, we can activate factor 10. From the intrinsic pathway, we can activate factor 10. And once you get factor 10 activated, it takes us through the process to actually end up making the fibrin. Okay. We say that this is a positive feedback loop because we're trying to increase the speed of this over and over and over again. The goal is get it done, okay? Not bring it back to normal, stop it, get it stopped, just like pushing the baby out, okay? Once we get the baby out, contractions stop. Same thing here. We're gonna keep doing this, amping it up as fast as we can so we don't lose very much blood, and then we're gonna stop it, okay? And then once we get it stopped, okay, we have to seriously stop it, okay? We don't want it to keep going. So we have to start releasing things that say, okay, we got to stop this. Heparin is one of those things that you hear about a lot. There's tubes that are heparinized, and basically that keeps your blood from clotting, okay? So when you go and have your blood drawn and they put it into a bunch of little bitty tubes, okay, they don't want it to be a big clotted mess. So we have heparin in these tubes, and that keeps it flowing, okay? And that's what we want to do here. Once we get the blood flow stopped, now let's get everything back to normal, okay? So those calcium ions that we talked about earlier, even vitamin K. Vitamin K is very important here in clotting the blood, okay? So, and oftentimes people that have clotting issues are given vitamin K, okay? It is essential in the clotting process. Once we get everything stopped, now the platelets are able to actually pull. They have been activated. Now let's deactivate them so that don't have the surface area, and let's pull them together. And basically that's almost like stitching the wound together. Okay, it takes anywhere from 30 to 60 minutes here. And while this is going on, okay, we've had enough time. Remember that cell division takes about 80 minutes. Now, from the time we cut ourselves, we've already got new cells that are coming online now, repairing this. So we need to start very slowly tearing down the clot, okay? So dissolve that clot, and the way we do that is there is a plasma protein called plasminogen, Okay. And that thrombin that is produced, along with there is a tissue plasminogen activator, it starts cranking up this plasminogen producing the plasmin. And plasmin's job is to digest those strands of fibrin. Okay. Remember, it's a protein, so it's going to be knocking out all those peptide bonds, digesting them down into the amino acids. And as it's going to happen as we are adding more cells. As we go through this process of fixing it, okay, we're going to gradually be tearing down the clot.